All right, so these are notes that I'm going to give you on Hardy-Weinberg evolutionary theory. So Hardy and Weinberg were two different people, two different gentlemen working in the early 1900s. And what they did was they actually, as many scientists do when they're really good at their jobs and they're really paying attention, is they blended what was already known, what other famous scientists had already figured out about genes and heredity and natural selection into one modern theory of evolution. So, for example, remember that Darwin's ideas were about natural selection. That was his mechanism that explained how populations could change. So he had natural selection, and they had read his papers on that, and the idea that the population is the unit that evolves. The population evolves. And remember that Darwin was doing that at a time when nobody knew anything about mutations or DNA or chromosomes. He was just using his observational skills to try to figure out how evolution might work. And then over here we have, well, he was working in like the 1820s or so. And then over here in the 1860s, we have Gregor Mendel. And Gregor Mendel's contribution to modern theory was his understanding of, right? He's the father of heredity. So heredity and his idea, he was the one that invented the term of the allele. Alleles that are genes, essentially, that control expressions of traits. He was the one that said there could be dominant versus recessive alleles or forms of the gene that could blend together into a phenotype. So also these ideas were ideas that Hardy and Weinberg used to develop their idea of Hardy-Weinberg, modern evolutionary theory. And they were working in the 1910, 1920s kind of era there. So maybe not modern, but still standing today. Their ideas that they came up with are still with us. So you can still call it modern because it incorporates all of the things, the genes. It also incorporates the idea of mutations and the genes and the chromosomes. So I'll kind of add that over here that there was, and we didn't really go over this guy, but there was a Thomas Hunt Morgan who worked with those fruit flies that you had a huge lab about. You may remember he did that fruit fly lab. Morgan was the one who came up with the idea of mutations and the idea that genes are located on chromosomes. Right. So that was also important to what they came up with, right? All of these things. And he was in he was in the early 1900s too. So early 1900s. Okay. So modern evolutionary theory, basically what Hardy-Weinberg said was that combining all these things, they came up with the idea that there is this thing called a gene pool. A gene pool, which consists of all the genes or all the alleles, right? Both dominant and recessive, in one population, for any one trait, right? All the genes for that one trait are like all grouped together. And that their idea was that evolution is a mathematical thing where evolution occurs when the frequency, which is something that you can calculate, of the alleles in a gene pool changes over time from one generation to the next. Change over time. So that's the basics of it. Now let's go into more detail. So the HardyWeinberg theory kind of comes in two parts. One part of it is that they made a list of conditions. And again, I will save this, and this is being recorded, so if you're not getting everything as I'm writing it, don't panic. You'll get to it. Okay. All right. So they said, there were two of them, they asked a question. They posed the question, what are the conditions? What conditions will cause allele frequencies to remain in equilibrium, to remain in equilibrium? In other words, not change, not evolve. Under what conditions would there be no evolution? So the first they came up with is that, well, there can't be any mutations. No changes in the genes, otherwise you have evolution, right? There can't be any natural selection. No natural selection. There is no order or no pattern or no advantage of any one trait over another one. Everyone survives, basically, with no direction in changing adaptations. No immigration or emigration, which is another word for that is gene flow. No gene flow. In other words, no immigration into the population or emigration, anyone leaving the population. That would change the population and that would change the allele frequencies, right? If you're adding or taking away to the gene pool. Mating has to be perfectly random. So no choosing mates based on how strong or fierce or how well they dance for you, right? Mating is random. Any kind of sexual selection might cause allele frequency change. And finally, the one that a lot of students forget, and that may be one of the harder, oh no, don't do that again. Oh God, don't do that again. Can we do this? Yes. At random. The population needs to be large. A large population is more likely to stay in equilibrium because the numbers, mathematically, the numbers are so large that small changes in whatever's going on, little bits of people either not surviving or surviving, won't change the overall frequency. Really tiny populations can have strange things happen to the allele frequency just from random chance. Okay, so those are the 5 conditions. Those are the 5 conditions under which evolution can't occur, which basically should tell you what? If you look at these, are any of these possible? No. Which pretty much was their conclusion, is that evolution is inevitable. Even if you don't have anybody leaving or entering the population, even if there's no natural selection, no mating is completely random, nobody's being picky about who they marry, mutations happen as a chemical fact over time. Accumulation of neutral mutations is automatic. There's really no way of stopping that. So evolution is kind of inevitable. But in any case, you can kind of point to reasons why a certain frequency change might happen that might be different from another reason, depending on the population and the situation. So now we get to the actual math. Math time. Okay. There are two HardyWeinberg equations. And they're actually not that bad. Pretty simple algebra here. Your first two variables. First two variables are E, which is the frequency of the dominant allele. In the gene pool, right? So we'll call that A. And Q is the frequency of the recessive allele in the gene pool. We'll call that little a. All right? The addition of both, sorry, P plus Q equals 1. So the frequency of both, assuming there's no other form of allele due to any weird mutations, is going to equal to 1. So for example, if you had a beaker, I used to actually do this with beaker up here, but I'm not going to do that today. And I put beans in the beaker, and the white beans were the dominant allele, and the black beans were the recessive allele. And let's say that they were exactly 50-50. So there were 50 big A's and 50 little a's. Then the frequency here would be P would be equal to 0.5, because 50 is half of 100. And Q would be the other 0.5. Therefore, 0.5 plus 0.5 would equal 1. And that's how those frequencies are expressed, in decibels like that. So there's that. Okay. The next part is, all right, so we have alleles, right? But remember that every organism, right, each individual has 2 alleles for each trait. To make up the genotype, there's a Mendel word, for each phenotype. This necessitates another equation. Oh, no, not yet. There we go. Okay. All right. So this is what they did. They basically expressed the crossing of an entire population's P's and Q values as a Punnett square, just as if we were doing a Gregor-Mendel Punnett square. So they put P and Q and P and Q to cross the P's and Q's of each generation. And if you do this just simple algebraically, right? P gives you P squared. PQ gives you PQ. PQ gives you another PQ, and Q gives you Q squared. And if you express then this as an equation, the second equation goes like this: P squared plus 2PQ plus Q squared, everything in the Punnett squares, again, equals 1. All right, So what do these mean? These are the genotypes that are possible with those A's and big A's and little a's. So this is the homozygous dominant genotype. This represents the heterozygous genotype. And this represents the recessive, homozygous recessive genotype, right? So for example, if you still have P equals 0.5 and Q equals 0.5, let's put that in here. So your double P squared, if we did that, would be 0.25, wouldn't it? Because 0.5 times 0.5 is 0.25. Your 2PQ would be 2 times 0.5 times 0.5, which comes to 0.50, right? And again, 0.5 times 0.5 gives you another 0.25, and doesn't that equal 1? Okay, I know I'm going a little fast, but overall, if you have a perfect gene pool that's in perfect half and half equilibrium, that's what you would have of your heterozygous versus homozygous dominant versus homozygous recessive individuals. That's what you'd get. Okay, so if you had no evolution happening, then generation after generation after generation, your P and your Q would stay at 0.5. And your percentages of individuals with these genotypes would stay the same, generation after generation after generation, except that almost never happens. So now let's try an actual problem that lets you practice this. A lot of times they'll give you something like this, just this right here. Let's try this problem right here if you can see it. In a certain population, the dominant phenotype of a certain trait occurs 91% of the time. Look what it says, the dominant phenotype, okay? And then it says, what is the frequency of the dominant allele? So first of all, you got to figure out, based on the variables I just told you, what is this 91% exactly? The dominant phenotype means that this is all the individuals, the percentage of individuals who are either this or that, right? So in other words, P squared plus 2PQ equals 0.91, right? So that's all that. That would be tough to figure out. You really can't figure out that. So what you need to do almost all of these problems is you want to figure out how to get to Q. And in this case, you can get to Q squared, can't you? Because if this equals 0.91, what will Q squared be? Because Q squared, according to the equation I just showed you, is the last part of this, right? 0.09, right? Yeah, that's it. Okay. That's a much easier place to start. So what we can say is, well, we can say that Q squared is 0.09. Okay, can I get to Q by itself easily? How do I do that? If I wanted to just solve for Q here algebraically, what would I do? Think back to your simple algebra class. Couldn't you do this? That. Okay, this is where you need your calculator and where I forgot mine, but it's okay because I have mine. Okay, so Q equals what? Tell me what Q is going to equal if you do that. I already did this. Isn't Q 0.3? Yes, thank you, Sophia. Good. Okay. So 0.3, not 0.03, but 0.3. Watch your zeros. Make sure you write those down correctly. Okay. So if Q is 0.03, aren't they looking for P? That's what they're looking for, right? The frequency of the allele would be, they're looking for P by itself, right? So what is P going to equal if Q is 0.3? And we know P plus Q equals 1. Come on. 1 minus 0.3 is 0.7. Thank you, Ryan. That's it. You've got it. This is the answer. This is the frequency of just the dominant allele, right? Because P is the dominant allele, and that's what they asked for, and there's your answer. Yes? Okay. Here's the next question right here. I want you to try this. Take a few minutes, use your calculator, figure it out on your paper, and I'll give you the answer once you've had a chance to work on it. See what you can do with this. Remember, always go for Q first. Here's the key thing that they're giving you. So I'm going to translate 4% into a 0.04 value, right? Which is the correct way to go from percent to a decimal. And this 0.04 value is the percent of babies who have sickle cell anemia. So it's this condition, right? And that condition, if you go back up to the equation, that means they're giving you Q squared. That's what they're giving you, right? So then let's just work our way down. If Q squared equals 0.04, solve for Q. Always solve for Q first because that gives you a nice tool. So your Q is going to be 0.2, yes? And therefore your P, which gives you your other important value, is 0.8. But that's not what they asked you for, okay? So now you have to look at what they asked. How many individuals, remember we've got 1,000 here, are you going to expect for each of the 3 possible genotypes? Okay, so genotype 1 is this one, genotype 2 is this one, and the third genotype would be this one, right? So this one's easy. You just take the original percentage they gave you, right? Q squared equals 0.04. And you multiply that times 1,000 to get the number of babies that have that. And you should get 40, right? Everyone with me so far? Okay. All right, so, yeah, all right, good. Okay, so the heterozygous condition, if you go back up to the original, I don't want to move it because I'm afraid that it's going to do something weird with the ink, but that would be 2PQ, right? To get the right percentage of the heterozygous condition, which equals, go back over here and use these, right? Which would be 2 times 0.8 times, oops, sorry, times 0.2, right? 2PQ times 1,000, right? To get the right number for the heterozygotes. And I get 320. You guys agree with me? Okay, good. And then the homozygous condition, yep, okay, good. I'm glad. Let me know, because otherwise I'm afraid I'm going too fast. All right, P squared is the last one, right? So you just take the P value over here, and you're going to do 0.8 squared times 1,000, because that's our population, and you should get 640. Yes? Does everyone agree with my results? Sometimes my tick is off, but I think I got this right. Okay, and this is your answer. This would be the answer that they would want right here. The number of individuals, right? They want the number of individuals that have each one of these different possible genotypes. Okay? Did you guys get it before I did? Or at least understand it as I did it? Either way, good. Okay. Now let's go down and thicken the plot a little bit. Now, what if, calculate a new allele frequency for just the dominant and recessive alleles. If in the next generation, out of 1,000 babies born, now we have 2% sickle cell anemia, 60% are carriers, and 38% are homozygous normal. I want to know if you can go backwards here. If you're really hot stuff, and you can go from the percentages I'm giving you and come up with a new P and Q. See what you can do with that. Yeah, now we have Q squared equals 0.02, right? So there's, the way most of you are probably going to do this is like this, right? And you're going to get a Q value of, what are you getting for that? Can someone do a quick square root for that? I left my calculator somewhere else.
0.1414.
0.14? All right. That's fine. Okay. So then what would P equal if that's true? I think it's 0.86. Isn't that right? Okay. All right. So according to this, you have a fluctuation in the allele frequency, yes? Except that I wanted you to notice something. Look at the carriers and look at the homozygous normal. What's the reason for that? Why are the carriers of this allele, why did their population go so much higher than the ones that were just homozygous normal? Wouldn't it be better to be normal? Yes. Immune to what? There's an immunity factor here. Yes. Okay. Remember that. And this is what? Remember that this condition, the heterozygous condition, gives you an advantage, gives immunity to a totally different parasitic disease called malaria. Yes. Okay. So there is something called the heterozygous advantage happening here. And I don't want to go into it too deeply, but you might be asked to explain, even without making a calculation, even without doing that, why is this larger than this? Why is being a carrier seeming to give you some stabilizing selection? And then finally, the last thing I'm going to ask before I stop this video is, which one of the 5 conditions of HardyWeinberg was violated here, which caused this change? All right, because the allele frequency changed, which of the 5 conditions? Yes, natural selection. Natural selection would be the 2-point value FRQ points for that question, right? And that is because, I'll just put it down here. Why did the carriers of the mutant allele do so well? Because they had an advantage and would have been more likely to survive to reproduce than either the sickle cell babies or even the homozygous dominant babies, because either one of those babies were vulnerable to malaria. They have no protection at all. So it's just a unique situation. Okay, but Hardy-Weinberg problems fit into that. So hopefully that was helpful in terms of introducing you to Hardy-Weinberg conditions and calculations. I'm going to stop this video